Hello everybody, and thanks so much for inviting me. I'm keen and trepidatious about sharing this work with you. It's drawing on the work that I've been doing in my British Academy Fellowship, and in particular the book that's coming out of that I'm writing with Martin Linley, who's a molecular biologist. And so I've given him a name check on this presentation because too much of the work that's come out of it is stuff that we've done together. So I'll be trying to challenge my molecular biologist while I'm here, and we'll see how I manage with that. I mean, I think one of the things to say as I start is that I'm still not sure whether it's a good idea what I'm doing, and I want to present 2 years of work where I've departed from what I've been doing and what people know me for doing, and increasingly set aside, I think, some of the key concerns that I held 2 years ago, and I'm still not certain that that it's, I don't want to say the right choice, but I'm still not quite certain where it's going to go, what it will produce for us, and whether we'll say retrospectively that it was worth the investment of my time and effort and energy. So it really is work that I'm doing. So purposes, I want to think about methodological and political implications of doing biosocial research, and part of that involves exploring some shifting conceptions of the human. I want to have a little look at the division between naturalistic and experimental methods, and in a sense I'm pushing the ethnography that I've done back into kind of an older school idea of naturalistic work, which probably 10 years ago I'd have said, no, that's not what I do. And I want to think about what biological and social methods can offer to building a biosocial ethnography. My key issue really is it possible to build biosocial ethnography, and what's it going to look like, and what's it going to be able to do for us? And in doing that, and maybe this is slightly tangential to that, but I want to think about some of the imaging and representations that biology throws up, and the way that becomes popularized, and the sorts of hopes and fantasies about the contemporary or future human that throws up. And to think about biosocial inquiry into feeling, and the feeling in learning and the feeling of learning. So my BA fellowship promised the British Academy that I would synthesize new biosciences and sociology of education to develop new ways of understanding the learner and learning. Big promise. And hopefully some of what's come out of that will be shown today. The book Biosocial Education is in process. It should be out early in 2018. And what we're trying to do is demonstrate what working across biology and sociology to think about education can do, and also where the difficulties lie, working through some of the tensions and the impossibilities that we encounter as we do that. Oh, Mac user. Okay. This, for me, is fundamentally about working transdomain, and what I found absolutely essential is collaborations with people in other fields. I don't think this is something that I can do as a sociologist without having, making friends with, or commandeering the expertise of people in analytic chemistry or genetics, or indeed science and technology studies. When I started this work, science and technology studies seemed to be quite a long way away from sociology of education. And of course, now that I've been trying to work with analytic chemists, science and technology studies seems quite close to sociology of education, the languages and the ways of thinking and the sorts of questions that are asked. So the work owes much, not just to Martin Linley, but to Jim Reynolds, who's an analytic chemist, Andy Bagshaw, who's a brain imaging expert, Sarabjit Matsana, who's a geneticist, we've been putting together research proposals together, thinking about what would it look like to do this, and who the hell are we going to get to give us the money to actually do it. So in one tiny slide, how I got here, I think how I got to doing this was increasingly taking seriously the desire in a new materialist turn to think about matter and to think about bodies and to think about them in ways that were more satisfying than I had been finding in the work that I had been doing and been drawing on, and finding really compelling the idea of assemblage. And I won't say much about it because it's probably what some of our most common ground. I have a hunch that's probably some of our most common ground. But the engagement with the bio seemed to me to be an obvious way of encountering those forces of assemblage when we're thinking about learner or learning. If I want to know how children feel, if I want to know why this classroom feels the way it feels, if I want to know what we can do here, then if I'm taking assemblage seriously, it seemed to me that it made sense to think about what were the bioforces, what might be the bioforces in play that were part of that. Other name checks we can come back to if we want to talk about them. So I think one of the things that has emerged over the course of the work has been a difficulty for me with pursuing this line of work without the idea of the subject. And I'm not letting go of the idea of the subject. I'm not letting go of the idea of a subjectivated subject. But the fields, the fields of social and political theory and feminism and sociology of education that I would normally locate myself in, and which I'm kind of departing from as I go and talk to my new bioscience colleagues, have increasingly been talking about the human and the post-human. And so one of the things that I want to do is to be able to articulate the work that I'm doing with that, but also to hold on to, at least as a productive force within this assemblage of learning that I'm increasingly concerned with, the forces of subjectivation. And so I hold on to Butler's work in Precarious Life about that concerns itself with the constitution of the human and the not human in the sorts of those sorts of old forms of subjectivation that people have started to call into question. And also to note that within her post-humanism, Braidotti continues to work with an idea of a subject. The subject doesn't disappear. It's another moment of degrounding the subject. So let's not let our subjects adhere too firmly in place when we're engaging with them as products of forces and as productive forces. I've been really influenced by Samantha Frost's work on biocultural creatures and the way in which she works through the instantiation of the social within the body. And of course, the ethics and the politics of this human presses on the work. And I think I've chosen to not say so much about that because of time, but I think that I'm anticipating that people's responses are going to be about ethics and politics. Some of this speaks to, I think lots of this speaks to the entanglements with the outside. And one of the things that I've more recently found very provocative, I don't know whether I'm going to continue to use it, but this work around the holobient that's coming out of the Human Microbiome Project seems to me to open up another set of lines of entanglement which perhaps we hadn't previously been thinking of. So if the Human Genome Project was looking for the genome to explain the subject, the Human Microbiome Project is looking for the way that the microbiota in and on the body influence the way that the body works, what the body can do, so that the genetic code of the human is only part of the story, and that very significantly the genetic codes of those microbiota are involved in the regulation expression of the human body. I found this all very exciting because it opens up the human to rethinking in yet another new way that I hadn't been anticipating. It's doing all of this without us even starting to have a conversation about epigenetics, and yet Schneider and Winslow, who've written much about this, that we seem in their work to still have a degree of wholeness in the human, while they talk about the plurality of the plurality of matter and plurality of code and plurality of influence. They ultimately say that experience trumps all of that in a sense, and that our experience of wholeness has primacy over that kind of biological multiplicity. I don't know that we need to that doesn't necessarily need to follow from the analysis. What I find provocative about it is the way that it really insists that we let go of our inside-outside distinctions. I was really hesitant to even name the inside and the outside of the body in speaking about it, but it seems difficult to let it go without talking about it first. But what I find really generative are these various lines of work that speak to us about the porosity and the plasticity of the body, whether it's Frost's traffic within biocultural creatures across membranes, Hannah Landecker's claim that we should understand the human as metabolic process. This microbiome project work about the integrated continuous whole of cells, which are human and microbial. The epigenetic claim that there is enduring and intergenerational interaction between the environment and the body's DNA, and the way that then genes are regulated and expressed. Beyond epigenetics, but within new biological sciences, there's lots of lines of work that look at the body and environment entanglement in nutrition, exercise physiology, the influence of toxins. They might not be arguing particular epigenetic effects, but they're arguing about profound influences. And I really find useful Malibu's suggestion that we think about transformability as ontological status. That offers to me a way of thinking about the human subject, the human person, that is still practical enough for me to hold on to, whilst keeping in mind these multiple lines of influence and of changeability. She talks also about the plasticity of concepts, which again has been a really compelling part of the work that we've been doing. That's demanded that concepts themselves are open for interrogation, and in some senses, I think that it's the concepts and the willingness to let go of the concepts that have been so fundamental to us, whether for me that concept is subjectivation, or whether the concept is woman, or the concept is learner. The possibility that those concepts themselves are put under interrogation and open to what Malibu calls a metamorphosis is for me really useful. So in coming to work across domains, some of the older work of mine with Judith Butler, where we've used degrounding, her thinking about degrounding as the place where we're looking to be, has been quite helpful. Elizabeth Wilson talks about a mutual rough and tumble and vertiginous alliances across disciplines. And I think that, I've been writing with Samantha Frost, and we've been talking about domains instead of disciplines as a way to try and get outside of the constraints of our disciplinary locations and to indicate the wider knowledge domains that those disciplines are part of. Elizabeth Wilson also talks about the suspension of a skeptic versus enthusiast position, and it's certainly something that I've felt really strongly in the work that I've been doing, that in different sub-areas of education, I find people who are hugely enthusiastic about neuroscience, for instance, and I find people who are hugely skeptical and to the point of absolute renunciation of findings from new biological sciences as they're being offered up to education. I want us to be really careful about what that offering up looks like and means, and who it's being done by, and to what ends, and what that project's about. Wilson says that we should bring to light a systemic traffic in disquiet that cannot be fully grasped by either, in her case she's talking about the neuroscientific or critical texts, but can be glimpsed at these moments of disjuncture where neurological data and critical inquiry meet and cleave across each other. So this is what I'm trying to do, not only to my own work, but also to encourage my collaborators to be willing to do with their work. One of the things that I think runs through the work in both some of the science and technology studies and in the new biosciences, and I think that resonates with some of the post-structural work and some of my own work, is this idea of a shift from things and names to things and functions. And in the microbiome project, the idea that we move from a need or a desire and ambition to name the microbiota that are part of the human body, to a need and desire to understand what the functions of those microbiota are, offers for me a reorientation for the work as a whole. And although of course the sorts of data that are being engaged and the sorts of questions being asked are vastly different, for me the social focus on processes and experiences and affects and assemblages and complexity aren't so far away from the mechanisms and functions and complex systems that some of my bioscience colleagues are trying to look at. We continue with this social science critique of laboratories and experimental models and a suggestion of reductivism, that scientific experimentation needs to reduce to a single variable to demonstrate a singular effect. And my experience is that whilst within a particular study or a particular experiment, that may well be what the model asks for and what my colleagues do, they do that with a strong sense of the relationship of that mechanism to further mechanisms. And I think that impulse to worry about the apparent reductivism of science is something that we need to be really mindful about where that takes us and how that relates to what our potential collaborators might actually be doing. Not to disregard, I've got policy love of RCTs in brackets, not to disregard the fact that at policy level they love RCTs. That's what we're being compelled to do. There's a lovely recent paper by Gary Thomas that tracks and takes to pieces this RCT as the gold standard in education research. So it's about understanding that those things are running simultaneously and that we need to navigate both of them. I think we also, maybe this sounds a bit like a bit of a confessional, but in thinking about doing experimental work, I think one of the things that it confronts me as an ethnographer with is another round of thinking about the social sciences relationship with the natural, and no matter what we might have said over many years of doing post-structurally informed ethnographic and qualitative research, the natural environment lingers in our work, and the natural environment in which the human subject is the most important thing there lingers in our work. And I don't want to make in any sense a sweeping desire to change that. I just want us to know that and make that choice purposefully, not accidentally or by default. But as we encounter the problem of taking biosciences out of the laboratory and into the everyday of schools, and may feel discomforted by the sorts of things that might mean that we're going to do or we're going to suggest get done in classrooms, I think it's important that we're mindful of the fact that we've been doing all sorts of interventions in classrooms for many years, that our research always intervenes in classrooms. We know that, and that if we have particular concerns about particular experimental methods from biosciences in classrooms, then it's important to be able to say why. Why does that raise a particular concern? I say this, it raises particular concerns for me, so I'm not, I don't want to suggest that it doesn't, but I want to ask why. Why does an EEG in a classroom make me feel discomforted in a way that a video recorder might have made us feel uncomfortable 20 years ago, but no longer does? So having those conversations, I think, is really important. So we have this myself and this group of people that I'm collaborating with, we have this ambition to undertake biosocial ethnography in classrooms to interrogate learning. One of the things that it raises in a way that hasn't been raised for me before is a question about the scales of granularity at which we're working. So, which potentially reach from a concern with capitalist global orders to discourses, to institutions, everyday life, to organs and affects, and cells and membranes, and genes and microbiota, and neurotransmitters and hormones, and that that to research those, not only are we interested in these different scales of granularity, but researching those different grain sizes takes different techniques, suggests different sorts of questions and problems. So this has implications for what we might seek to see or feel or know or understand about a classroom or about learning. It has implications for methods, it has implications for analytics. I think some of the implications for analytics we're only just beginning to scratch the surface of, and I'm hopeful for the day when we've got the kind of fundamentally incompatible data that we want to analyse simultaneously. At the moment, we're still writing protocols. So what are some of the options and or enticements that we might engage and draw on. So I want to talk a bit about seeing the inside. So of course we have the human brain, the organ. We have early, I'm sure probably you're all quite familiar with early attempts to identify the relationship between areas of the brain and their functions. So here we're in phrenology, we're concentrating on the surface of the brain. With I think one of the things that really strikes me about it is the effective and moral tenor to some of the things that are identified. And they're identified through losses, through surgeries, right? So things like secretiveness and veneration and hope as located in different parts of the brain. We go on to contemporary region functional correspondences, and suddenly all of those sorts of things get contracted down into just the limbic association cortex down the bottom there. So we have a different sort of account. The practical functions that are absent in phrenology are now what's primary. I wanted to share this with you. So sorry if it starts to seem like I'm just really enjoying it. Some of these things I do really enjoy. Let's see if this will work for us. This is, here we go. The very latest scan of the wiring of the human brain. Notice that this brain is wired, that the wiring metaphor persists. Note the music of wonder and amazement. Look at everyone's face, wow. It's so beautiful. The image that we're looking at is CGI. We're not looking at the scan, we're looking at what the scientific animators have done with the data. One of the reasons that I show this is because I think that there's a fundamental question of whether we know the difference between the data and the CGI footage, and whether the people who generated the data can say very much about what the data tells them about function. And again, that metaphor of wiring, kind of old school first generation computers, still persists in the popular account of the brain. I want to show you similar things in another way. So here's a schematic of the chromosome and unwound. It's unwound through the chromatin, and then you can see the actual DNA and the histones that they're spooled around, and the way that they're spooled around is part of regulation. And then you can also see coming in epigenetic factors, the little orange pentagon that attaches and changes what the DNA on that histone can do. So we've got this this schematic of the whole process of epigenetic influence. But of course, what we actually have, if we're looking, the microscopy of the chromosome is quite different. Under massive magnification, what we see is a really complex structure. What we don't see is the schematic that we just looked at. This is even greater magnification. It's a magnification of a mouse embryo, very early development of a mouse embryo. So the top row is just under light microscope, and then the second two rows are they've got they're images of staining, so they use fluorescence to stain and to bring out particular things. So the red line is we're looking at the methylation of a particular protein, lysine, and then on the next row we're looking at the DNA. What we see is that the schematic again doesn't show us what the bioscientists see. The schematic shows us something else. And then if we want to go further, let's see if that's set up in precisely the place I want it to be.
What I am going to show you are the astonishing molecular machines that create the living fabric of your body. Now,
Okay.
molecules are...
Let's see if I can take you to where I want us to be.
By tiny, I mean really. They're smaller than the wavelength of light, so we have no way...
It was supposed to be set up to play exactly where I wanted it to be. Of course, okay, at about 5 minutes, I think.
This was work from a number...
That's...
This is what I want to show you next is updated science, it's updated technology. So again, we begin with DNA, and it's jiggling and wiggling there because of the surrounding supermolecules, which are stripped away so you can see something. DNA is about 2 nanometers across, which is really quite tiny. But in each one of your cells, each strand of DNA is about 30 to 40 million nanometers long. So to keep the DNA organized, regulate access to the genetic code, it's wrapped around these purple proteins. I've labeled them purple here. It's packaged up and bundled up.
So labeling colors?
A single strand of DNA. This huge package of DNA is called a chromosome. And we'll come back to chromosomes in a minute. We're pulling out, we're zooming out through a nuclear pore, which is sort of the gateway to this compartment that holds all the DNA, called the nucleus. All of this field of view is about a semester's worth of biology, and I've got 7 minutes, so we're not going to be able to do that today. No, I'm being told no. This is the way a living cell looks down a light microscope, and it's been filmed under time-lapse, which is why you can see it moving. The nuclear envelope breaks down. These sausage-shaped things are the chromosomes, and we'll focus on them. They go through this very striking motion that is focused on these little red spots. When the cell feels it's ready to go, it rips apart the chromosome. One set of DNA goes to one side, the other side gets the other set of DNA, identical copies of DNA, and then the cell splits down the middle. And again, you have billions of cells undergoing this process right now inside of you. Now we're going to rewind and just focus on the chromosomes.
So notice he said when the cell felt it was ready to go.
So again, here we are at that equator moment. The chromosomes line up, and if we isolate just one chromosome, we're going to pull it out and have a look at its structure. So this is one of the biggest molecular structures that you have, in, at least as far as we've discovered so far.
So watch this moment. This is the moment where we go.
A single chromosome, and you have
we go from
DNA in each chromosome. One is bundled
a view through a microscope to CGI, and we just transition.
These whiskers that are sticking out from either side are the dynamic scaffolding of the cell. They're called microtubules, but the name's not so important. But what we're going to focus on is this red region. I've labeled it red here, and it's the interface between the dynamic scaffolding and the chromosomes. It is obviously central to the movement of the chromosomes. We have no idea, really, as to how it's achieving that movement. We've been studying this thing called the kinetic core for over 100 years with intense study, and we're still just beginning to discover what it's all about. It is made up of about 200 different types of proteins, thousands of proteins in total. It is a signal broadcasting system. It broadcasts through chemical signals, telling the rest of the cell when it's ready, when it feels that everything is aligned and ready to go for the separation of the chromosomes. It is able to couple onto the growing and shrinking microtubules. It's transiently, it's involved with the growing of the microtubules, and it's able to transiently couple onto them. It's also a tension sensing system. It's able to feel when the cell is ready, when the chromosome is correctly positioned. It's turning green here because it feels that everything is just right. And you'll see that there's one little last chip that's still remaining red. And it's walked away down the microtubules. That is the signal broadcasting system, sending up its stop signal, and it's walked away. I mean, it's that mechanical. it's molecular clockwork. This is how you work at the molecular scale. So with a little bit of molecular eye candy, we've got kinesins, which are the orange ones. They're little molecular courier molecules walking one way. And here are the dynein. They're carrying that very broadcasting system, and they've got their long legs so they can step around obstacles and so on. So again, this is all derived accurately from the science. The problem is we can't show it to you any other way. Exploring at the frontier of science.
I'm going to skip his, the big moment where he's going to save the world as well. I wanted to show you that because we have this moment. He says what he's doing is he's showing us the unseeable, and he's using CGI to show us what cannot be seen, but that is there. And we have this moment where we move from a view down a microscope into CGI that kind of happens completely unannounced. And we have this anthropomorphized cell that feels things and knows things. And we have this metaphor of machines that runs through these little machines that live on the microtubules and that have legs and they walk up and down. And of course, they don't do any of those things. They didn't feel anything. They didn't know it was time. It's a attributing of a rational thought as an agentic subject. This that's not there. But of course they are moving. They do have there is a propulsion mechanism, and they move. It's just they aren't legs, and they didn't think now's the time. This is the little grey box. That's a kinesin at a magnification of 10 nanometers, which is like a factor of 100 of magnification. So if you can see the little white squiggle, and then on the far side you can see a schematic of the kinesin. So on the one hand, isn't it delicious that we can move from this to the CGI moving representation of that kinesin doing its job? But at the same time, we need to understand that that is a CGI image, and that the scientists don't know that it's there and what it's doing by watching. They know probably by blotting for the presence or the absence of the protein. They know it's happening chemically, not visually, but to help us. So on the one hand, it's a nice way of talking to people, but there are lots of little clips like that. TED have quite a few nice ones. There's one by another scientific animator who was commissioned by Harvard to make animations like that would be the core of their undergraduate biology program. So it's not just about helping people who are not molecular scientists understand what's happening. It's also about a new way of conceiving of the discipline from within the discipline. The one I'm thinking of in particular has God music in the background, which was particularly lovely. So I'm sharing those because I want to illustrate some of the sorts of science that are on offer to us as, for me, as a sociologist of education who wants to think about learning, wants to think about the way that the body at the molecular level might be embroiled in those processes of learning. But that's only one set of tools, and they're a set of tools that are at some distance from what we might ordinarily do, or what I might ordinarily do. So I also want to put alongside this some of Anna's work around feeling, which there's a range of people who've done work on feeling. I think Anna's is really particularly useful wherever she is. She's in the room, so I have to just say nice stuff about her. So what speaks to me in this work when I want to think about what the bioscience can offer me is this very careful distinction that she makes between affectus and affection and affect, and that we're going to tease apart the feeling experienced by the body and the changes in bodily capacities that may be associated with that. And the idea that feelings might be a means for researching affectus, and for understanding of how, in Anna's work, how arts makes embodied shifts in capacity. But for me, it may well not be arts that make that shift, but it's that shift in capacities and how we might research them and how we might know about them. So she talks about a focus on affective assemblages in research contexts, smells and sounds and aesthetic economies, indirect discourses. And these are the sorts of things that in much of my work in school trouble, for instance, we see me struggling with trying to speak to those phenomena and what they make do, what they might make happen. And now in relation to the bioscience work, I've been looking at what other ways might we encounter the affective in our research. So I've been looking at different biosocial approaches. I think Celia Roberts' work that she talks about biopsychosocial, and as Martin and I have gone on to write about feeling in the classroom, we've ended up with this kind of endless flow of interrelated types of influence: socio-cultural, psycho, biochemical, techno, and all of these flows and functions entangled with each other. What we want to be able to do is talk with specificity about the nature of those entanglements, to be able to say we are convinced of these entanglements, we're convinced that they are simultaneously chemical and molecular and social and cultural. What does that look like? How does that work? As we start talking about how we're going to do that then, and I'm likely to take up the position of the ethnographer primarily, the bioscience collaborators then start offering their potential ways in. We start finding that we're talking about the sorts of structures and processes that we want to identify, and they're vastly different. So we might want to identify structures and processes biochemically, or we might want to identify them discursively, or we might want to identify them effectively. And these infer different questions, but they also infer different methods. And it's that understanding how they're entangled that we keep coming back to. So Fmri scanning, I'm sure people have seen images like this before. Happy brains and sad brains. It's just areas of the brain which have enhanced blood flow in response to happy stimuli and sad stimuli. This is EEG of brain connectivity during emotional stimulus. This is a sort of work that Andy Bagshaw does. He does not so much on in response to film provocation, but this is a sort of measurement that Andy does. So he will use remote wireless caps which have got pickups on them, a number of pickups, and we have conversations about, do you want 32 pickups or do you want 64 pickups, and it depends on what sort of connectivity you want, and it's harder to put them on the children, and how are the children going to feel wearing them. And I find myself in we're currently writing a bid where we're going to have a number of children at once in a classroom with EEG while they go about their daily business. And so they're really practical, are kids going to tolerate as many pickups as we want, and how much movement will the system be able to tolerate? How normal can their practice in the classroom be? But we think it's worth trying, even if it crashes and burns, because what this sort of literature demonstrates is that there are patterned responses that the EEG can pick up that appear to be associated with different sorts of emotional states. So we want to see if we can make that work in a classroom, and whether we can find anything, whether it suggests anything useful for us for thinking about the way that affectivity is allowed in classrooms. Here's another one that we're working with. Jim Reynolds in our team is an analytic chemist specialist in volatile organic compounds. This is a piece from a different researcher who's taken atmospheric VOC measurements in cinemas. So this is the audience watching the Hunger Games, and what the science does, it collects samples from the atmosphere, and they're timed samples. And then what it does is it measures the mass of the ions within the compounds within the sample. And by measuring the mass of the ions, they can work out what those compounds are. So what they've got is CO2, and then they've got acetone and isoprene. And they are suggestive of energy production, which is suggestive of enhanced emotion, maybe. In this paper, they make an association with isopropyl acetone as metabolites of cortisol. Some of the analytic chemists are, more or less convinced by that. You can't take a sample of the atmosphere and just say, this is from these chemicals, these are the feelings that are circulating in this room. But what we might be able to do is say something more tentative about the affect of a space. It works also, we also do breath samples. So Jim's done work where 12-year-olds have taken tests and have done individual samples before and after the test. And what those samples show, no surprise, is how stressful the testing is. We did expect them to find it stressful, and they did find it stressful. It's another way of coming to that analysis. And when my Facebook feed is covered in articles from teachers and parents about children whose eyelashes are falling out because they're so terrified of doing their SATs, I wonder whether there might actually be some mileage in us taking some measurements of volatile organic compounds in classrooms during testing regimes as a way of giving some further life to our arguments about what classrooms do feel like and what classrooms might be able to feel like if we treated the children in them differently, or we invited them to engage in different sorts of practices. Now, I know that there's a danger there that sounds like I'm saying that I want my big friend in science to come and help me win an argument. And part of me doesn't care. Like, as a practical politics, I think there are moments where I don't mind. And even if that only rewrites how big and how strong my big friend in science who's come to help me is, if it's been an effective line of argument, then it might be worth it. But I think these are some of the pragmatic political choices that we might need to make. But what we aren't suggesting that we're going to stop doing ethnographic work with children. What we're suggesting is that we might do ethnographic children, which includes sometimes that we take the portable mass spec into the classroom and we all have a bit of a laugh while we blow into it. This is, I'm finishing. This is from a piece that me and Jane Kenway wrote in the introduction to a special of Emotion Space Society on the emotional geographies of education. And it's an account of a boy in the tree-lined lawns of a special school running away from some teachers. I'm I'm considering whether I'll read it. I'll let you read it. You've probably read it by now, have you? When Jane and I picked this out of my field notes and were delighted by it, we wanted it. We felt that it went to the affection of schooling and of these sorts of school spaces, and that what we could see in the data were the flows of feeling through the people involved in this moment. And we make that case. And I wonder, though, would we make a different case? Would we make the same case? What if we all blew into the mass spec at that moment, or in the moments before? Or what if we'd had any one of us had had an EEG in the moments or the hours before? What else might we be able to understand about this boy's running, about where he runs and where he doesn't run, about what any of that might mean educationally? So I wonder about the way that working across these domains forces us to think again about our concepts, learning, the disorder of the child in that school, the feeling or the affectus that we're interested in understanding. I'm interested in the ways that it might allow us into this idea from Catherine Malibu of change as an ontological status. We were convinced about the malleability of the subject. The subject was never fixed. But does this now invites us to think about that malleability within biochemical as well as discursive limits. And it also allows us to think about the locus of that malleability being produced through and productive of the entanglements of the biochemical and the discursive. So I had two questions that I wanted to pose. I'm no closer to the answer at the end than I am at the beginning. Should social science move from critique to engage productively with biosciences, as I've been trying to do? And can biological and social sciences really collaborate on transdomain empirical research, in particular, in my case, in education? Okay, done.